Hi, the topic of our project is the evolutionary significance of introns by Mio Zimba and Mariama Garba. Introns. Introns are non-coding segments of an RNA transcript or the DNA encoding that are spliced out before the RNA molecule is translated into a protein. The coding sections are known as exons. Alternative splicing. Alternative splicing, a regulated process during gene expression that results in a single gene coding for multiple proteins. A gene is first transcribed into a pre-messenger RNA, pre-mRNA. A copy of the genomic DNA contains introns, destined to be removed during pre-mRNA processing, and exons, destined to be retained within the mRNA in order to code the protein sequence. As you can tell, introns were immediately and universally dismissed as genomic debris. However, introns have evolved from the notion of just being junk DNA. In this video, we will discuss the evolution leading to designed DNA. The fact that all higher organisms possess introns and the more complex organisms possess a higher proportion of introns indicates that they serve at least minor and possibly major functions. Carrying this enormous extra baggage of unnecessary DNA each generation is a heavy price to pay. What could explain the loss of introns in lower organisms? Variety is critical for species survival, and producing variety is difficult in animals with small genomes. Putative ancient bacteria evidently would possess little capacity for variety. If the absence of introns in modern bacteria was also the situation in early bacteria, the presence of introns in eukaryotes and their absence in prokaryotes has prompted three different theories regarding the initial origin. This figure showcases the tree of life. The origin of introns as explained. The green branches indicate lineages containing introns, the black branches denote pre-intron stages, and the red branches indicate secondary loss of introns. The intron late theory suggests that introns and the spliceosome arose within the eukaryotic lineage. Then the introns accumulated in eukaryotic genomes. The intron's early theory states that intron-exon structure of genes was present in the last universal common ancestor and possibly earlier, where primordial protein domains were shuffled to facilitate protein evolution. The intron first theory is similar to the intron early theory, but proposes that introns and the spliceosomes are remnants from the RNA world. The model was initiated from the observation that putatively ancient snRNA genes are often encoded by introns, because RNAs were the only catalyst for the assembly of an all-RNA ribosome before the advent of proteins. snRNA must have been used for the assembly of the proto-ribosome as it is evolved towards full protein producing capacity. The introns that contain snRNA predate the protein coding exons that surround them. The splicing of snRNA encoding introns from transcripts with no protein coding potential and the processing of pre-RNA and pre-tRNA by RNase-P are examples of how RNA processing might have occurred before proteins evolved. What is the evolutionary significance of introns? The definition of introns has evolved from non-coding segments to a complex mix of vital DNA. This figure showcases the origin and evolution of intron retention. Mutations of DNA sequence, including point mutations and sequence deletions or insertions, have the possibilities to weaken the strength of the splice sites and change the conditions of splicing, which is making the normal introns retained. The retained introns might originate from exonic sequences. Point mutations on the exonic sequences have the possibilities to create weaker splice sites and introns to be retained. This image showcases the mobile elements of other DNA fragments and that it can be integrated into a proto-splice site, location of thus creating introns, and it is possible that some of these new introns may be retained. The inverted red triangles represent the mutations of DNA sequences. Gray box represents the normal intron, while the green box represents introns that are retained. White boxes represent exons. Intron retention leads to regulation of gene expression and increase in protein diversity. Nested genes. The discovery that some DNA functions as exons when expressed by one pathway, but as introns when expressed by another. It blurs the distinction between introns and exons. Research by Oliver Smithies of the University of Wisconsin showcased that introns may have an important role in controlling the expression of exons. DNA functions as exons or introns, depending on the pathway leading to enhancement of transcription. It is also possible that introns have a role in reducing or correcting mutations. That brings us to the role of introns in cancer. Studies have shown that introns could be directly or indirectly involved in cancer causation. Shan and Heichel concluded that inheritance of intronic polymorphism in p53 gene increases breast cancer risk. Despite the detrimental possibility, introns may contribute to a greater genetic diversity due to their role in crossing over. The main source of genetic variety in eukaryotes are introns that possibly assist in the recombination process to make sure that exons are shuffled in an orderly manner to produce appropriate combinations. What question is next? Introns have evolved from junk DNA to design DNA, DNA that may be critical for life. Much is still left to be learned about the significance of introns. Does the presence or absence of introns have any clinical relevance on human health? Learning the impact could be a better understanding of cancer through introns.